is one for which you could get multiple distinct phenotypes from just one single genotype due to differences in environmental conditions. So the butterfly that we used was Bicyclus lemoniana, which is phenotypically plastic. In the wet season, you have this butterfly with very conspicuous eye spots, while in the dry season, you have reduced or absent eye spots, and the butterfly is very cryptic. Another less obvious plastic trait is their sexual ornamentation, which is this UV reflective white pupil. And this is also plastic because in the wet season, males have more UV reflectance relative to the dry season males. And also the dry season females have higher UV reflectance in this pupil relative to wet season females. And this trait is accompanied by a really interesting behavior, which is sexual reversal. So in the wet season, you have what you would expect, which is males court females, and females choose mates based on the UV reflectance of this pupil. But in the dry season, you have the females courting the males, and the males court females based on presence or absence of this UV reflective pupil. And so this was really interesting as a behavior, and since we study vision, we were wondering what is happening to their vision systems. And a previous study looked at their eyesight, because in studies in flies, we know that maintaining vision is very costly, and flies that are kept in captivity for a long time have decreased their eye sizes. So they looked at the different sexes and at the seasonal forms, and they found that males have larger eyes, so they're sexually dimorphic for eye size, and they're also plastic because wet season forms have larger eyes relative to dry season forms. They also looked at opsin expression using qPCR, and they looked at the UV opsin blue and long wavelength, and opsin is the protein that finds a chromophore and through phototransduction converts light into an electrical signal. And what you do see is this trend where dry season males and wet season males don't have different expression in their opsins, but females have a significant downregulation when they're not exhibiting a choosy behavior. And so we would expect that if there is relaxed selection on non-choosy individuals. So what I wanted to see was what other genes are associated with these two morphologies. So what other genes are differentially expressed between these sexes or the seasonal forms. And I hypothesized that phototransduction genes, in addition to the opsins, other ones would follow this trend where they're upregulated in choosy wet season females. And Aide developmental genes would be differentially expressed between individuals with different eye sizes. So for example, probably upregulated in males with larger eyes and in wet season forms with larger eyes. And for the methods, I extracted RNA from 3 dry season females, 3 wet season females, 3 dry season males, and 3 wet season males. And I used Illumina TrueSeq to make 12 RNA sequencing libraries. Then I used Trinity to make a de novo assembly for this species. And I used RSEM to map the libraries back to the assembly. And I had a coverage of about 50x, and 85% of my reads were back into the assembly. So then I used EDGAR for a whole transcriptome approach to find differentially expressed genes. But I also, since we're interested in vision, we did a candidate gene approach, and we did 2 ANOVAs on 203 candidate genes. So to find the function of our contigs in the Trinity assembly, I used transdecoder to find open reading frames, and I did a blast to Drosophila to find Go terms and homologous genes. So these are our results. NDE is differentially expressed contigs, and GO is gene ontology terms. So for sex, we find 290 differentially expressed contigs with 77 unique Go terms in Drosophila. And we found 722 differentially expressed contigs per season and 111 for their interaction with sex and season. And so it's like very semantics for seasons. So we wanted to explore what the function of these genes was. So we did a gene enrichment for these 229 gene ontology terms. And we found functions in cuticle structure, also alpha-tocopherol transport, extracellular region, juvenile hormone binding protein, sugar transporter, and protein dimerization activity. So that's kind of interesting, but the one that we were really looking at was this one here because it functions in alpha-tocopherol transport, and a gene in Drosophila known as Pinta also has this function, and it's used to transport the chromophore. So this is one that is probably having to do with vision. And in addition to those gene enrichment, we also did a visual inspection of each of the genes individually, and we found 2 eye developmental genes differentially expressed by seasons, which are CLAR and warts, and 1 eye pigment biosynthesis gene, henna. So now looking at the candidate genes, first we looked at the opsins just to validate the previous results. And as you can see on the y-axis, that is FPKM, which is a measure of expression. These are very highly expressed. The long wavelength gene was the 3rd most highly expressed gene across libraries. And we don't see that the opsins are differentially expressed between types there. But in the blue, we do see that there's a significant difference between dry season females and wet season females, and between dry season females and wet season males. We also see this is true in the UV, where dry season females are downregulated relative to wet season females. And we would expect that because these dry season females are not exhibiting the choosy behavior. So we see downregulation in non-choosy dry season females. We then looked at the full transcriptome to see what genes are probably co-expressed, so have similar expression to the opsins. And we found this contig, which also has this alpha-tocopherol transfer activity, which I mentioned earlier might be a putative gene transporting the chromophore, so important for vision. And it has this Krell trio domain, which in our lab recently was found to have lineage-specific expansions in Lepidoptera. So for our candidate gene approach, this one we did 2 ANOVAs for sex season and the interaction of sex season, and we found 33 genes differentially expressed for eye development, 8 for bone transduction, and 8 for eye pigment. And I'm not going to talk about all of these, but we did think it was interesting to have so many in the eye development, and we wanted to see how these are regulated. We thought that maybe genes in a similar pathway would be regulated in a similar manner. So I put these into gene networks for key eye developmental signaling pathways. EGFR, JAK-STAT, wingless, armadillo, notch, and hedgehog. And what you can see, it's like a lot of information, but what you can take away from it is that key eye developmental genes are conserved between Drosophila and butterflies. You see that there's differential gene expression between sexes and seasonal forms. So the green boxes are upregulated in wet season, and the blue ones are upregulated in males, which we did expect to see. And we also see that genes are shared between signaling pathways. So not all of the genes within a pathway are going in the same direction, but they are influencing each other. So some of the conclusions are that we find opsin genes, an eye pigment biosynthesis gene, and a transfer gene downregulated in non-choosy dry season females relative to choosy females. We also see that 2 eye developmental genes are differentially expressed between seasonal forms. And we see that genes in eye developmental networks have varied expression in sexes and seasonal forms. So we suggest that genes regulating developmental pathways are good candidates in driving divergent eye sizes. And there's a larger effect of season on females on vision-related genes. So with that, I'd like to thank my lab, my committee, my collaborators, and my funding sources. And thank you for listening.
have time for some questions if anybody has any.
Yes?
What tissue are you using for your RNA extractions?
Okay, that's a good question. We use